Hello, I am Dakota Kalin. The information located in a gene is used to construct a product in the process known as gene expression. A gene that will encode a polypeptide is expressed in two different stages. During this process, DNA leads to RNA, which in turn leads to proteins, a relationship that has come to be known as the central dogma. The first step in the central dogma is transcription, which is composed of three stages: initiation, elongation, and termination. It starts at the start point and typically extends several dozen or more nucleotide pairs upstream from the start point. RNA polymerase binds in a precise location and orientation on the promoter. In eukaryotes, transcription factors mediate the binding of RNA polymerase and the initiation of transcription. Elongation. As RNA-P moves along the DNA, it unwinds the helix and exposes the DNA nucleotides for pairing with RNA nucleotides. The enzyme adds nucleotides to the 3' end. As the mRNA is made, RNA polymerase rewinds the mRNA. Termination. For eukaryotes, RNA polymerase transcribes a terminator sequence in DNA. Then, mRNA and polymerase detaches. It is now called pre-mRNA. In eukaryotes, cells modify RNA after transcription. The 5' end is synthesized first and receives a 5' cap. The 3' end of the pre-RNA molecule is also modified before the RNA exits the nucleus. The poly-A tail of about 50 to 200 A's are added. These additions serve three functions: facilitate the export of mature mRNA from the nucleus, help protect the mRNA from degradation by hydraulic enzymes, and help ribosomes attach to the 5' end of the mRNA once the mRNA reaches the cytoplasm. RNA splicing is a stage of RNA processing in the eukaryotic nucleus that involves the removal of large portions of the RNA molecule that is initially synthesized. Most eukaryotic genes and their RNA transcripts have long enough regions that are not translated. The non-coding sequences are called introns, and the coding regions are exons. RNA polymerase II transcribes both introns and exons, from DNA. But the mRNA that eventually enters the cytoplasm is abridged. The introns are cut out and the exons that remain are joined together, forming an mRNA molecule with the continuous coding sequence. Small nuclear ribonucleoproteins recognize splice sites. They are joined with additional proteins to form a splicosome. This interacts with the sites along an intron and removes them while joining two exons that were next to the intron. The second major step is translation, where proteins are made from RNA. Translation is a very complex set of reactions that take mRNA and turn it into proteins using a multitude of enzymes and, most importantly, tRNA. First in the process, a tRNA molecule arrives at the ribosome with code for a very specific amino acid. This is at one end of the tRNA. At the other end of the tRNA is a nucleotide anticodon. This anticodon is what gives the ribosomes the code to produce the amino acid chains. The tRNA acts as a translator for this process. It is the step used to connect the code found in mRNA to the proteins produced in the chain from the ribosomes. The tRNA used in this process, just like the mRNA, is produced in the nucleus. tRNA is only about 80 nucleotides long, which is shorter than most mRNA chains. There are three major phases to translation. The first step is initiation. In this stage, the ribosome comes around mRNA and the start codon is attached to the first tRNA. Translation cannot begin without the start codon at the 5 cap. The basis for this start codon is AUG. The next step of translation is, of course, elongation. This is where peptide synthesis really takes off. In the ribosome, tRNA attaches to the mRNA chain in order to synthesize different amino acids and create an amino acid chain. tRNA enters the ribosome molecule by molecule, while the mRNA is fed through the ribosome like a chain that attaches to each tRNA molecule. This process moves very quickly in order to synthesize long amino acid chains that will be sent out into the cell later. The final major phase or step in translation is called termination. In termination, one of the 3 stop codons enters the ribosome. tRNA molecules do not bind to these stop codons and therefore are then hydrolyzed. The peptide on the tRNA is just released into the cytoplasm and the ribosomes dissociate from the current chain of mRNA. The ribosome then gets ready for the next translation process, thus ending the translation process that was first taking place. After the amino acid chain is released, it then begins to contract and fold into itself. This process is called protein synthesis. The single amino acid chain itself folds to form the primary structure, which then continues to form the other protein structures thanks to the help of different enzymes. Each of the amino acid chains that are synthesized by all the parts in translation are then sent out into the cell to accomplish all sorts of different purposes. Some of the proteins synthesized are even sent out of the cell to fulfill other purposes. The proteins synthesized by this reaction can help catalyze other reactions or help give structure to the cell.